let me show you, or let me share you a pipeline that we have set up in the server tbioinfo platform. Pipeline is actually designed to compare, contrast, and benchmark different clustering methods using gene expression data. So to give you a very short overview about gene expression data, we all know that this data contains vital information that is required to understand the processes that takes place in a biological organism in relationship to its phenotypical conditions or characteristics or environment. So to understand and deciphering these hidden patterns in this gene expression data will help us strengthen our understanding of the complexity that is buried in the biological networks, and the general complexity of the gene expression data or omics data and the volume of the genes present increases our challenges of comprehending, interpreting, and analyzing these huge mass or volume of data. So the use of data mining techniques like clustering is one of the first few steps towards addressing these challenges. And we need to perform these analysis methods to process several patterns that are buried and interesting patterns that are underlying in the data. So let's go back to the pipeline, and I want to bring to your notice to the sequence of analysis that is involved in this pipeline. So we start with the principal component analysis in our library, and then we follow it up and we take the output of the principal component analysis and then apply a multitude of clustering techniques like hierarchical clustering, like k-means clustering, spectral clustering, DB scan, etc. And we finish we end the pipeline. So we do this to compare these clustering methods interpretation of the patterns within the gene expression data that was provided as an input. So before running to the output files, let me tell you or briefly show you what the input looks like. So if you check this input, this is an expression data, and this data will look like something like this as a table. This is a structured data that Ilya had introduced you to, and this data is organized in such a way that the samples are in the columns and the genes are in the rows. Usually, this data will be of a huge dimension, right? The dimensions of the gene can vary anywhere between tens of thousands to even more, like 20s of thousands. So if you check this one, we have curated this data, and we have around 7,000 genes in this data. Even this is big data, right? So to understand the patterns of this big data, what we actually do is we perform principal component analysis before clustering. So what PCA does is, PCA is actually used to project this data of huge dimensions into a lower dimensional subspace, and then the clustering is applied in this subspace. So using PCA before clustering actually logically reduces the or decreases the computational cost, as we are actually implementing clustering on the reduced dimensions of the data. PCA will actually provide distribution of the data points in the transformed dimensional space, but PCA itself will not cluster the data points. So to demonstrate you, let's explore some of the outputs that you find in the pipeline. Once you run such pipeline, you will get some visualization options. And if you take some of the visualization option, I think I have already loaded this visualization. So let's go there. Yeah, so you can explore the principal component analysis output of this pipeline in a 3D interactive plot. So this immediately shows us there is some really good separation between the samples of different types. And if I can move this over, yeah, and we have the color coded legend also available. So we quickly understand that there is a very well or good separation between the samples, or at least between the luminal and non-luminal type, and we can employ clustering to observe whether these patterns are actually in the clustering techniques or not, or do we see any other surprising information in the clustering. So if you check another output, which is the tabular output of this principal component analysis platform, then we will see that the dimension, which is of 7,000 genes that we have inputted, had been compressed to 3. Here we have given only 3. It could be depending upon the setting, 3, 5, 10, or how many components that you are going to calculate, right? So thus a decrease, or using clustering on this decreased dimensional space will actually speed up your process, and also will eliminate the unwanted noise which could be present when you are dealing with the high dimensional data. So to understand all of this pipeline, we will have several of these examples in the session or in the program. But now I want to point you or focus on the bottleneck of this pipeline, which is the principal component analysis on our input data. So if you click on the output files, you will get outputs that are associated with all the analysis that we have run, we have actually used here, included here. And if you include, or if you download this principal component analysis _r pack.zip, and this actually contain the code that was necessary for the principal component analysis to generate this output. So let's go there. Let's take it out. Yeah. So this pack contains the input file as we had downloaded, and I showed it to you, and it contains the standard R code with which the server actually ran this analysis. So let's, yeah, this is the R code which the server used to run this principal component analysis pipeline. But we have given, or we have dissected this R code in a step-by-step manner in an associated lesson, which you get a free chance or a chance to explore only for today. So if somebody can paste the link, let me find the link myself. Yes. So this is a lesson that deals with the principal component analysis of your input data. And I give you the link for this lesson in the chat box. You are free to open this lesson and explore the codes along with me, and we can walk through the different components of the code, and we will have a separate space where we are going to apply this code and run this code from the Learn OmicsLogic platform themselves. Yes. Okay. So in this lesson, we start with an introduction to what principal component analysis can do and how it is very useful for us to first explore the data using principal component analysis as such. And after you have passed through that, we give some examples of the input methods or input table. Then we discuss this table now, and we will also discuss this table in greater detail during the program. And in the Learn the Code section, this lesson is organized by first giving you an introduction or a specific aim for what this lesson is intended to teach you or explain you. And then we straight away jump into Learn the Code section, where first you get a demonstration of how to use PCA for your data. And then we have a code breakdown. So what do we call as code breakdown? We break down the codes into fundamental components of the code and explain the relevance of this code and explain how it is, or how different components of the code and the syntax of the code come together and help you perform this analysis. For example, we have to load some of the libraries which will enable you to run this analysis in, for example, in R's environment. So it is usually done by including the library function and followed by the name of the library. And then we definitely have to load our input file, and this is done by defining where the file is, or defining the location of the file using this syntax. And this is the assignment operator in R, which will take the value of whatever is on this side of the operator and then assigns to the object that is on this side of the operator. And then we load up the table or load up the input file using a read.table function. And read.table function takes at least one parameter, and that one parameter has to be the location from where you can access the file, right? This location can be your local computer, or this location can be a publicly available link that we usually share with during these lessons. And we have to describe how our data looks like, right? At least for a few of the parameters, we have to describe how our data looks like. If you check our input file, our input file contains, if I can go there, yes, our input file contains the first row, which tells you what each and every column is supposed to be. So, or in other words, in programming languages, the first row is the header of the columns, right? We know that this column represents the expression value of the sample that is named by named this code, and so on and so forth. And we need to mention that to R so that the data is actually loaded correctly. So that becomes one of the description of our input file. And we have several other descriptions, like how two columns are separated. That will form the separator that separates two columns in our data. This separator can be a tab, this separator can be a comma, or this separator can be any special character that you can use, like semicolon, like pipe operator, and etc. And you can also mention some additional parameters, like how you want to treat the characters that is present in your data table, and whether or not you want to load the first column as the row name, which will be very useful for further purposes, right? And then once you have loaded the table into the object full underscore table, which in this case, the read.table function loads this as a data frame. So what is a data frame? You will learn much more about data frame and etc. In the sessions. So in a nutshell, data frame is a collection of data of various types. A matrix or an array is a collection of data of specific data types, but while data frame can have data of different data types, like a character, like the logical character, like categorical character, and numerical character, of course, numerical inputs, of course. So in order to run several of these statistical analysis and several of these analysis, we may need to convert our data frame into a matrix format so that the numbers are interpreted properly. So we do that by converting only those part of the data frame which contains the numerical information, and we convert that into matrix, and we store that into a matrix by the name expression. And then we have to apply principal component analysis along the genes as dimension. So if you see, or if you notice our input data had the genes across rows. So we need to transpose the table, and that's what this specific code does. And then we can, now our input file is ready and data file is ready to be analyzed for principal component analysis, and we can invoke or we can call the application, or we can apply principal component analysis in R by calling the function prcomp. And again, prcomp at its basic level takes at least one input or one parameter that has to be the matrix that for which the principal component analysis has to be calculated. And we mention that matrix, and in addition to that, you can mention other options like how many components you have to calculate, or you can mention other components like do we have to scale the data, do we have to center the data, etc. And once you run this analysis, then you can store the result of this analysis in this object PCA. And there are several options in R that will leverage the different organizations of information that is present in this PCA object, so that we can plot, we can explore, and we can understand the principal component or analyzed components of our input data. So at the basic level, we can actually plot the principal component analysis first two components, right? PC1 and PC2, we can simply plot it, and we can understand that it produces some good separation of samples. Right? But this is just to see that whether we have any outlier in our sample, or how do we go about interpreting our data. So this is very basic plot, which gives us very little information. Like for example, in this case, we see that there are no alarming outliers in our data, and there is some sort of grouping that is observed, but without labeling these groupings or without annotating these samples, we don't know what these where these samples are, what these samples come from. So that is what we can do in the following plots, where we explore different options, or exploring the range of data visualization options and parameters, so that you can label, you can color, and increase the amount of information that you can extract from this principal component analysis pipeline or output. So just like using plot, we have another way of interpreting the PCA, which takes GG45's library, and autoplot is that function, and that function comes with added advantage of having some presets that are very useful to us. Like, it takes the percentage of variance explained by principal component 1 and principal component 2 automatically, without the necessary of explicitly defining there, and it produces some really nice appealing graphics. And in addition to that, what we can do, we can expand, as I mentioned, to other options or other parameters in this autoplot function to include the labels for the sample, and we can color the labels, and we can color mention the size of the labels, and that will add one more layer of information that we can that will help us understand the pattern. Now we can see that how where individual samples are actually located in our principal component analysis. This is useful, very useful, if you have only a handful number of samples. Even in this case, we have something around 40 or 52 samples, and it becomes really difficult to read some of them and to understand what that sample means, or what this group means, or what this specific sample mean, and what are the characteristics or phenotypical characteristics or different biological conditions under which the measurement from these samples are taken.